Welcome to everyone here already. We'll just wait another minute or two to get started. All right, good morning and welcome to today's APS event entitled Insights into Forebrain Control of Breathing. I'll be serving as your moderator today. My name is Brian Gelbach, and I'm a member of the respiration section and the webinar programming committee. I'm joined by my co-moderator, Dr. Alexandra Babacheva from the Hormel Institute at the University of Minnesota. In today's session, Dr. Brian Dlouhy will present insights into forebrain control of breathing from human intracranial electrophysiology studies. A few housekeeping notes up front. Out of respect for our speaker, we ask that you refrain from photo or video captures of the presentations in whole or in part. The session will be recorded and available for future viewing through a link on the APS website. Taking photographs of another person's presentation without their explicit permission is prohibited. At this time, all attendees except for the speaker are in listen-only mode with video off. As moderator, I'll be happy to raise questions that are entered into the Q&A function on the bottom of your screen. Please use this function instead of the chat. This session will end at approximately 12 p.m. Eastern Time. It's now my pleasure to introduce our speaker, Dr. Brian Dlouhy. Dr. Dlouhy is Associate Professor at the University of Iowa Hospitals and Clinics. He's a practicing neurosurgeon whose research lab is dedicated to exploring how the forebrain controls and responds to breathing. Dr. Dlouhy's primary focus is on people with epilepsy who are undergoing intracranial electrode monitoring to pinpoint the origins of their seizures. These patients and procedures provide unique access to forebrain structures that modulate breathing. As an occasional collaborator with Dr. Dlouhy, I've had the opportunity to see firsthand how powerful this approach can be, especially in his and his team's hands, and have learned a lot from his research. So with that, thank you, Dr. Dlouhy.
Thanks, Brian, for the welcoming introduction. Can you hear me?
Yes.
Perfect. Don't want to go through 60 minutes of just
That's a bit disappointing.
All right. Well, thank you so much. Yeah, so I'm honored really to be able to give this presentation today. Something I'm very passionate about. As Brian talked about, the title of today's talk is going to be Insights into Forebrain Control of Breathing from Human Intracranial Electrophysiology Studies. It's really been a pleasure working with Brian and learning a lot about physiology of breathing from him. And he's been a key collaborator with us ever since the beginning of this. So I have no disclosures, just funding from the NIH. When I would say that I work at the University of Iowa, and when I would go around and give talks and meet other people, I always tell them, from Iowa, I live in Iowa City, and here's our campus here, the University of Iowa. It's a beautiful campus. And people usually ask me, where's Iowa? Is that the potato state? Getting us confused with Idaho. But nowadays when I go, everyone says, "Oh, you're at the University of Iowa. You must be a big fan of Caitlin Clark," and that is what we are known for now. I would say everywhere I go, this is what I get. If you don't know who she is, she was last year a senior at the university and record-breaking year, and now a superstar and probably one of the best college women basketball players of all time, and now really becoming one of the best professional women's basketball players probably of all time. So, but I'm associate professor at the University of Iowa. I'm a neurosurgeon, and one of my focuses clinically is on epilepsy and surgical epilepsy, and trying to treat patients that have intractable epilepsy. That means they've really haven't been able to have their seizures treated even with medications. And so then they, if they're the right patient indicated for it, then they undergo surgery. And one of the surgeries that we do is placing electrodes into the brain. And this is we call it intracranial EEG, but it's an incredible high resolution tool to identify the seizure focus within the brain. And then if we can identify it, then we can resect that area of the brain. And so this is an intraoperative photo of electrodes that on the left there, and the 3-year-old girl with electrodes going in through the scalp into the brain. This is a procedure called stereo EEG, and it's a purely depth electrode approach. And here's an X-ray showing that. We get a CT scan after surgery, X-rays, we get an MRI, and to kind of really define where these electrodes are. And so there can be up to 250 electrode contacts recording from the brain at all times. And what we've developed here at the University of Iowa, this is true for our adult and pediatric epilepsy patients, is we have the patient room here on the left. And then there's this window here that's connected so you can look in and to our research room. And in the research room here's on the right, it's where all us as researchers are and where all the equipment is. So the patients don't have to see any of this. And as you know, breathing is very affected by many things, and it's affected by, people that might have some emotional or anxiety from seeing lots of equipment or things that may be going on. So really, this has been important for us, and I think going forward for these studies, and the hospital's obviously been very generous in creating this. They basically take a patient room and created this research room for that, for our purposes of these experiments and tasks that we really run in these patients. And these are, two patients, and just showing you kind of what they are. They have a head wrap on. Electrodes are all kind of connected together. They can get up, move around. They can move around the whole room. They can't leave the room, of course. The idea is we want them to have seizures so we can record them on the intracranial EEG, identify their seizure focus, and then hopefully, cure them of their epilepsy. But during that period of time when they're in the hospital, they're awake and interacting with us, we can record from their brain and undergo all these different respiratory experiments and tasks. So there's a little background of kind of how, my research lab and what we do. And I work with many people to do this. So it's not just a one-man effort here. Breathing, as we know, affects many things. It's important in something called sudden unexpected death in epilepsy, which I'll talk about. That's SUDEP or SIDS also, sudden infant death syndrome. And we know that breathing affects and can cause anxiety, and is affected in anxiety disorders, as well as anxiety disorders can cause breathing abnormalities. We know it's involved in sudden cardiac death and learning and memory and speech, sleep apnea, primary lung disorders, and even primary sinus disorders affect breathing. And the one thing that, we'll be focusing on, part of today and most of the day is really sudden unexpected death in epilepsy. And that's how I got really into, studying breathing control. And really how that got started was really, there was this unknown of, this how seizures cause loss of breathing. And that was really it was tied to SUDEP, because we knew that SUDEP, was really and over time is really thought to be this loss of breathing during seizures or from a seizure. We call that apnea. And it's very common in seizures. It occurs in 1/3 of all seizures, frequently seen in children. They turn blue or turn purple. And it's been observed since 1899 by a neurologist named Hughlings Jackson. We also know that in observed and witnessed SUDEP cases, we see respiratory dysfunction. They have apnea, and they're prone face down in bed. So there's an environmental component to it as well. But that's important because we know that that's probably also limiting airflow, and also, again, suggesting that, the big problem with SUDEP is breathing control and loss of ability to breathe. Often occurs in sleep with really no apparent struggle. So there's no alarm that kind of goes off. A big study that is really critical and identifying apnea and loss of breathing as a major cause of SUDEP was this international study over many years, decades, in fact, of patients that had died, epilepsy patients that had died in epilepsy monitoring units around the world. Now, this is an uncommon event. You never want this to happen. But in these cases, they were able to record breathing by just the video and EKG to look at heart rate and rhythm. And what they found was that apnea preceded asystole in all these monitored SUDEP cases, suggesting it wasn't a cardiac event really that was causing their death, but really it was a breathing issue, loss of breathing, and really it was a central apnea. And really, again, how these seizures stopped breathing was unknown for really over 100 years until we began studying our epilepsy patients undergoing these implanted electrodes to determine their seizure focus. Again, as I mentioned, this is intracranial EEG. This is another method that we can use. And some patients still undergo this, although I'd say it's more common to have the depth electrode only approach. But this is where you actually do a craniotomy, you open the covering of the brain, you place electrodes on the surface of the brain. And then here you get the patient's own, 3D rendering of their brain from CT, MRI, reconstruct this, and you can see where the electrodes are actually placed in the brain. And this is all kind of playing on their clinical indications and where we hypothesize seizure focus is coming from. Again, we're monitoring their breathing during their stay, and so many of them, all of them will have a respiratory belt on the whole time. That'll be a chest or abdominal belt, as you can see here on the right. And then, depending on how well they can keep it on, and for all of our kind of experimental tasks, they'll have this oral nasal thermistor that they wear here. As you can see, normal breathing's here. It's very sinusoidal, very oscillatory. And we get all, these 4 channels. And then you can see what is, this apnea or loss of breathing. Central apnea is what you see here, where there's this complete loss of airflow through the nose and mouth, and there's no movement of the chest or abdominal wall. So, what did we find? Well, we found that central apnea occurs when seizures spread to the amygdala. So this is one of our first patients who's 26 years old. And we found the right frontal cortex was here, the seizure focus and the seizure onset. And you can see this kind of very large spikes that are occurring here. You can see the breathing is unaffected. And that's the case, I would say, for most of the brain, that breathing is really unaffected. The patient's not moving. And what happens is when seizures spread to this left amygdala, and the seizure activity kind of begins right here. You can see it's kind of faster activity. You can see the apnea starts. And that's a central apnea, right? There's really no movement of the chest or abdominal wall, and there's no airflow. This lasts for about 47 seconds or so. Oxygen discharge to around 87% via pulse ox, and then the very end, breathing occurs toward the end of the seizure here. We saw this in this patient every single time the seizure spread to the left amygdala. And interestingly, every other seizure in which they did not spread to the left amygdala, we did not see any kind of apnea. Again, suggesting this was something that was more specific to the amygdala and not other locations in the brain. And that's a great, association, but it didn't prove cause and effect. So one of the tools that we can do as neurosurgeons is we can electrically stimulate these different contacts in the brain. And we can stimulate every single contact in the brain. And we do that as a mapping procedure. It's a really clinical procedure to identify sites that could be seizure foci, as well as to understand the function of the brain. And so I do this, and this takes a long time in these patients, sometimes a whole day or even a whole full 2 days to do this, but it's incredibly informative from a clinical perspective, but also it's useful from, research perspective. And in this case, we wanted to know, well, could this site and that we see the seizure spread to in the amygdala, could that also, is that really the cause of the apnea, right? So show cause and effect rather than just an association. And so electrical stimulation of that left amygdala caused immediate apnea, very, very similar to what you saw kind of previously here with the seizure, right? Almost identical to that. And we stimulate for 24 seconds. Apnea lasted the entire time. Then afterwards, breathing slowly kind of resumes over the next 10 or 15 seconds. You can see oxygen desats around 90%, as it did during the seizure. So we all know the brain has a left and right hemisphere, and many functions can be lateralized depending on the dominance of the hemisphere, as we'd like to say. But we want to know if there was this effect was lateralized to the left side, the dominant hemisphere in most people, or if it also the right amygdala could also cause it. So we stimulated the right amygdala, and we saw a very similar effect. Normal breathing, then just pure central apnea. and then a return of breathing a couple seconds later. Again, oxygen desaturation about 90%. It's delayed here because of a pulse ox. That's a peripheral monitoring. Presumably, this is all occurring during their loss of breathing, the oxygen desaturation. Now, to show this wasn't some widespread effect, throughout the, electrical current spread throughout the brain or to adjacent sites, we simulated just lateral to the amygdala, and this was about 5 to 10 millimeters, 2 contacts, and it had no effect on any breathing at all. You can see oxygen saturation was maintained, again suggesting this was an effect specific to the amygdala and not current spread. So we studied epilepsy patients, but we were also able to take advantage of studying a patient without epilepsy. And we studied this patient in the operating room during an awake craniotomy. So the patient's awake. We do surgery, open up take the bone off, open up the covering of the brain, and we're moving a tumor in the temporal lobe, and we do this for language mapping purposes. And when we got to the amygdala, we stimulated the amygdala and found that apnea occurred as well. And so these findings then suggested to us that this was not some effect that was specific to epilepsy itself or to recurrent seizures, and that this is likely an effect, a function of the amygdala in all of us, right, as humans. And really, epilepsy is just hijacking the amygdala. And seizures are hijacking the amygdala to cause the apnea during seizures. What was really striking to us was that patients were unaware of this apnea, and they lacked air hunger. And really the perception that they even stopped breathing. Here you can see this is even just 47 seconds of central apnea. There's one single breath here in the middle, and then afterwards, breathing is just kind of slowly comes back to normal and regular, but there's no hyperventilation. The patient's completely unaware of this. You ask them, they state that we didn't notice anything that was different during that period of the stimulation. They didn't know that their breathing changed. They really feel no effect. And this is despite, even oxygen saturation dropping, to 87%, and presumably a CO2 rising, right, which gives you that, oftentimes the CO2 drives that perception of breathing loss. Now, if I asked you to hold your breath at 47 seconds, you probably struggle to do that, or even 20 seconds. We asked this patient to do that, actually. We asked the patient to hold their breath for 20 seconds. They couldn't even do that. And then over time, they're able to do it for up to 60 seconds. And by the time they get to the end of this, the patient's red in the face, they're struggling to breathe. And you can see what happens afterwards, what you typically would expect, hyperventilation, right? This increased respiratory rate, big breaths, and oxygen saturation, again, dropping around, 91% or so. But you can see the big difference between, stimulation of the amygdala and just a volitional breath hold, right? So very, very different response afterwards. So we've studied in total now 76 adult and pediatric patients to date, all with similar findings, even as young as 6 months old, which is one of our more recent patients, a very, very unique case that we'll be presenting in the future. This is from our study in 2020, kind of showing, this how kind of specific this appears to be to the amygdala. And we did this study to see if, number one, if we could see it in children, as we saw it in adults previously, but also we did it to see if we could find a site in the amygdala, specifically that controlled the breathing and caused this apnea. But here's a great diagram showing you all the sites that we stimulated in this cohort of 8 subjects. So it's 174 sites in the brain and 342 stimulation trials. And all the gray bars are this period of stimulation. The red bars are the duration of apnea that occurs. And so a couple of things. You can see the apnea is almost immediate in every single case with the stimulation. So it's not some delayed effect. It occurs immediately. And you can see that when we got to lateral to the amygdala or the hippocampus, a site that's just adjacent to the amygdala, gyrus rectus, cingulate, or the frontal temporal parietal cortex, sites that all would think would be involved in breathing control or breathing perception, did not cause apnea. And so this data suggested to us that, hey, that there's this really unique function in the amygdala. The unique thing in this study as well was that we use these very closely spaced electrode contacts. So this is number 1 and number 8 contact, and they're all sitting within the amygdala. That's the MRI, and B here is showing you those are the actual size of the contacts. So they're very, very small, and they're spaced across the amygdala. The amygdala here, we parcellated it out in this individual patient's anatomy and the different nuclei of the amygdala. Because we know the amygdala has all different nuclei and different functions. And then we stimulated across those contacts and these different nuclei. So stimulation of 1 and 2 did not cause really any apnea, maybe a little blip here. And then just literally, a millimeter or 2 lateral to that in contacts 2 and 3 in the basal medial and basal lateral amygdala, we saw apnea. And then it got a little bit more pronounced when we went to contacts 3 and 4, lasted a little bit even longer past our stimulation, and then shortened up a little bit, but still the full length of our stimulation in contacts 4 and 5. And then a little bit shorter in contacts 5 and 6 in the lateral amygdala. And then, the basal lateral amygdala, and then the lateral amygdala even shorter. And we got to the very lateral aspect of the lateral amygdala. It was just a blip here of a loss of breathing. And again, suggesting that, some of these sites, maybe the more medial sites in the amygdala, and the basal medial, basolateral amygdala, had a greater effect in causing apnea than other nuclei. And so to do that across all subjects, we use a template brain from MNI, the Montreal Neurological Institute, to map all the sites so we could kind of see exactly where across all subjects it was causing the apnea, or in this case, no effect or no apnea. You can see all the red here was this kind of medial portion of the amygdala. And kind of overlapping this, we see is the kind of the cortical median nucleus, the amygdala transitional areas, and this kind of medial basal nuclei. And this is kind of a face-on view here on the left, and then a top-down view. You can kind of even better appreciate the red here causing apnea. All the white here is really no effect. You can see that back into the posterior part of from the anterior to the posterior part of the hippocampus. The black, which will be called transient apnea, and that was just apnea did not last the length of the stimulation. And so you can see there's this kind of graded effect from no apnea all the way to apnea here in the medial amygdala. Then we use machine learning to take all of the sites and the results of the stimulation trials, and we identified this area in the amygdala that inhibits respiration, and because it inhibits respiration, causes apnea, we call it the amygdala inhibition and respiration site, or the air site. And so it's a probability map on a template brain showing it. And then we mapped it onto again, a 3D template brain showing the amygdala here in this gray structure, the tan structure of the hippocampus. These are all different views. And this red site is the air site. And this is just a kind of a video and kind of 3D showing that. So this is the site that's the highest probability of causing apnea during stimulation or during seizures. And so that was great. That really kind of helped us kind of solidify that the amygdala is important for causing apnea and causing apnea during seizures, which I mentioned is a very common phenomena. So 1/3 of all seizures has apnea during the seizure. But SUDEP is really a post-ictal, and this after a seizure ends phenomenon. And going back to that study across all the patients that were done in these kind of the video EEG, epilepsy monitoring units, the seizure ended here, and this is after the seizure ends. And there's still some breathing, although breathing may be abnormal at this point after the seizure ends. The apnea that really led to the death occurred after the seizure ended. And it's presumably that there's probably some apnea that's occurring even during the seizure, like I just, and presumably structures like the amygdala was involved in that. But really the question at this point then we had was, well, what's causing this apnea after the seizure ends? Is the amygdala still involved in that? And so that was the big question for us the last couple of years. So what really underlies the post-ictal apnea that seems to be really critical for SUDEP? And what we found in a couple of patients when we kept studying, all of our patients with epilepsy and intracranial EEG and electrodes was that stimulation induced seizures in the amygdala. In some patients, elicited both ictal and postictal apnea. So this is an adult, and the red contacts here where we stimulated on the right, and the red contacts here we stimulated on the left. And on the right side here, we induced a seizure. So with stimulation, you can stimulate where you can be below seizure threshold, and you can stimulate above seizure threshold. And so it's changing the current or changing the parameters of your stimulation. In this case, we did that. And this is an important clinical tool, as I mentioned before, to kind of help say, well, is that a site that may be more prone to seizures? It's also helpful for us from a clinical research tool to see what happens to the patient as well. In this case, we're recording breathing and have a nasal airflow. Pressure here, you can see when we stimulate, we cause apnea. And then after the stimulation, a seizure is evoked, and you can see that the apnea kind of persists. There's some breaths here. And then after the seizure ends, you can see that there's still periods of apnea. Again, one of these bars is 30 seconds. So we're seeing, 5 or 10-second periods of apnea that last for a couple of minutes after the seizure ends. And you can see almost it starts getting a little bit longer here and longer. And then, for things for understanding we don't know yet, but still trying to figure out, is then all of a sudden breathing starts to become more regular again. The apnea shorten, and then you begin kind of back to baseline, regular kind of sinusoidal oscillatory breathing. But again, this was one of our first patients that we found this in, suggested to us that, wow, so in some patients, wow, the seizure in the amygdala may induce not only just ictal apnea, but also postictal apnea. It means after the seizure ends, apnea may still persist. So we saw that in additional patients when we do seizures. We also saw that with just pure electrical stimulation below seizure threshold. So this is an adult patient, where we stimulated the L2, L3 contacts. And you can see apnea occurred here, another period of apnea. There's some abnormal breathing here. And then after the stimulation ends, again, the patient's totally awake. They have these long periods of apnea. This lasts for about 2.5 minutes until finally breathing becomes a little bit more regular here, a little bit more regular, and these apneas shorten and shorten and shorten, and finally they get back to this baseline breathing. Here I'm showing you the oxygen saturation to show you that every single time this happens, oxygen saturation drops below 90%. So this was, again, showing that not only it was during these seizures that we could evoke, but also we could actually cause that. So this was showing just cause and effect as well, that not only the amygdala inhibiting breathing during seizures, but also after the seizure ends. And we could map this across patients, right? So we had numerous patients that had both, that had this kind of persistent effect of apnea during amygdala evoked seizures or stimulation-induced, just pure stimulation. And we found in this magenta color sites here that causes persistent apnea. The red was the sites that cause apnea. And then white is, again, no apnea. And again, it kind of clusters in this kind of anterior medial portion of the amygdala. Again, these very, very similar nuclei in the cortical median and amygdala transitional areas and medial basal nuclei. And then we use machine learning again to see if we could localize this to a more specific area in the amygdala. And interestingly, it lies within the air site, as you kind of predict from just the electrode context I showed you in the previous slide, but wholly within the air site and kind of in the very middle portion of meso portion of the amygdala. And because this site persistently causes inhibition of respiration, we call it the P air site, the persistent amygdala inhibition of respiration site. We are also able to do studies not only just while they're awake, but also in the OR. And we do these experiments. We can do them when they're awake, of course, doing awake craniotomies, and to look at kind of do different respiratory experimental paradigms. But we can also do it when the patient's on a ventilator, or at least under anesthesia with a breathing tube in place. And then it gives us this more precise respiratory measures. We can do stimulation in the operating room. And we do this when the patients are about to get the electrodes removed. So this is the end of their monitoring period. We've identified their seizure focus, and we've identified a surgical plan of what we're going to do. And they're going to essentially go home the next day. Before we do that, obviously, we have to remove the electrodes. But before we do that, we're able to record from their brain in the operating room, and we use this equipment here on the right. You can see that we can stimulate the brain here, as you can see. We can also use all the manipulations of the ventilator as well to understand how breathing is affecting the brain. This is a really very powerful tool for us, as researchers, as neurosurgeons, to kind of understand how the forebrain is affecting breathing. We use that tool in one of those patients that caused that where amygdala stimulation caused persistent apnea. And what we found there was that not only did the amygdala stimulation induce this kind of persistent apnea after the stimulation ended, but also induced hypoventilation and persistent hypoventilation and hypercapnia for many minutes after the stimulation ended. And that really suggested to us that the amygdala is, the stimulation of the amygdala is reducing CO2 respiratory drive. We're also able to do other experiments in which, while we're in the MRI scanner, while the electrodes are still in place. We all know about, functional MRI, and that's good, but it's really more just with resting, MRI, fMRI, you get kind of just simple chordal associations, whereas electrical stimulation with fMRI, we call ESFMRI, really is looking at these kind of causal connections between brain sites. So we can stimulate the amygdala, and then just immediately after that, then image the brain functionally and look at the bold signal and see what areas of the brain are affected by that stimulation. And so this is a very powerful tool for us because we don't have electrodes everywhere in the brain in all these patients, right? And one area that we don't have never have is the brainstem. So we want to see what was happening in the brainstem as well as other sites without electrodes. So what we found in one of those patients that had, again, the persistent apnea from stimulation and from seizures is that the amygdala stimulation decreased fMRI bold activity in the pons and medulla, as you can see here, and increased the bold activity in the insula. Again, this site in the brainstem, we think is, critical for, the brainstem control of breathing, the automatic control of breathing. And we know that the insula is critical for the interception of breathing or the perception of breathing and our perception of air hunger. Again, suggesting to us that the amygdala may be inhibiting breathing by affecting medullary respiratory network, the Preboscinger complex, may also be affecting then the sites that are sensitive to CO2 and the CO2 drive to breathe, such as neurons and the respiratory, and the RAF-A as well as the RTN, and may be affecting and disrupting the normal perception of breathing and the typical air hunger we perceive of loss of breathing by affecting the insula. So our conclusions from these studies was that we identified really a focal circumscribed area within the amygdala. We call it the P air site. Not only induces ictal apnea, but also persistent post-ictal apnea and persistently inhibits dyspnea. And this might be critical for SUDEP. We've identified this connection or circuit that exists between the amygdala and brainstem respiratory network, and we know this is thought to be an inhibitory pathway. That bold signal there in blue was inhibitory, and the red was thought to be more activating. And you can see in the brainstem it was blue, suggesting it was inhibitory. We also identified a connection or circuit between the amygdala and ventral insula that may be involved in the perception of loss of breathing. And importantly, the P air site, the changes in this may be predictive of SUDEP. So anatomical changes, volumetric changes, structural changes, molecular changes. This could be a potential therapeutic target for SUDEP as well. So how do we put this together for SUDEP? We have an epilepsy patient here. We have an amygdala and seizures that spread to this air site. The air site may cause then ictal apnea, and then they get recovery of breathing. And we see this very commonly in patients with seizures. Again, 1/3 of all seizures will cause this. Seizures are spread then to this kind of P air site, the persistent amygdala inhibition of respiration site. Not only do they have ictal apnea, but they get a persistent post-ictal apnea. And then again, this leads to this persistent air hunger. There's no alarm that goes off. And So the patients aren't calling out to their loved ones, they're not calling out to their parents, right? They're not trying to change their position. They don't know that anything is different. And so this kind of two-hit kind of hypothesis we have of what causes SUDEP, this loss of alarm, loss of air hunger, and this kind of persistent apnea. Interestingly, we think that, why do some patients have this, area in the amygdala, the P air site, and maybe others don't? First, I'd like to say we don't totally know. Maybe everyone does, but it's more likely that there's been structural changes that occur in the amygdala over time, maybe from repeated seizures. There may be genetic differences between individuals that lead to this sub-region of the air site that then causes this persistent effect that you see here on the right at SUDEP. So one question, that we've been trying to ask is, why do we as humans have a site that inhibits breathing and suppresses dyspnea? This may be for defense mechanisms, both involuntary or volitional, such as the typical freezing fear response many people have, studied, and the amygdala is critical for that. So, no, loss of breathing then allows you to camouflage yourself better, like the deer in the headlights look, the mice that freeze from different stimuli, fox odor, for example, et cetera. If you're not moving your lungs, right, less movement, maybe less ability to be able to be observed in the wild. And so maybe defense mechanism, protective mechanism. For us, breath holding, right? That's critical for many things that we do, important for protection of the airway. We don't want to breathe in different conditions, different gases that may be that are out there. So toxic gases and so forth. Also underwater diving. We as humans obviously do this. Many other mammals have this ability. Dolphins especially, right? And this can be trained, right? We can kind of train this. And one question is the amygdala, involved in that? Is critical for this? Maybe there's some data in the literature that maybe suggests that. There's also some data in the literature that maybe suggests in speech, to do what I'm doing now to maintain proper ventilation during speech, maintain, this kind of, prolonged speaking without taking a breath. CO2 levels rise during this time. And some experiments would suggest that that rise of CO2 would drive your ventilation so high that it'd be difficult to speak. And the amygdala may be important for that. And there may be many other things that's important for eating, playing wind instruments, even emotional states, laughing, crying. I think other things that we need to do that we have to hold our breath and suppress any kind of air hunger. So as I kind of mentioned, with SUDEP, this ability to detect our breathing, the interoception of breathing is really critical for survival. And so the interoception of breathing is this ability to continuously monitor and sense or perceive our own breathing. We know that disruptions in breathing patterns really demand immediate attention. Otherwise, oxygen saturation drops, and it just all SUDEP, right? You lead to eventually arrest. And breathing interoception influence, we also know that breathing interoception not only is critical for survival, but also influences emotional control and cognitive performance. And there's been some key studies looking at this in the literature in humans. And we've been trying to ask those kind of same questions and take the lead from others on this as well. And this is some data that we have that's almost finalized, and our submission is just immediately forthcoming here for this manuscript. I'm just going to show part of this data. But a big question for us is how and where does our forebrain respond to breathing? How does our forebrain, again, perceive breathing or even the loss of breathing? And how does that change even over time? So this is a template brain showing electrodes, a combination of both depth electrode only approach as well as a case where we had grids and strips and depth electrodes in. There are about 1,200 contacts total here. They're recording from in the brain. And the participants in this kind of these tasks, these are epilepsy patients still, but these participants are undergoing just resting breathing in this experiment. So they're just asked to breathe quietly while they're in their bed. And we have a respiratory belt on, we have nasal thermistor, oral nasal thermistor in place, and we're recording from each of these contacts the local field potential. So that's just the electrical activity around each of these contacts in the brain while we're recording their breathing. And then we can take these different sinusoidal brain wave patterns that we see and the local field potentials. And we can take the typical sinusoidal pattern of breathing that you see, and we can look at their coherence. And that's just a measure of looking at kind of how these oscillatory patterns, the breathing and the brain waves, how they kind of synchronize, at the breathing frequency. So this really kind of low breathing frequency that we all have as humans. And each of these sites, you can kind of see the significant sites that we saw, right? So these are the most significant sites from different coherence, from 0.2 to 0.8 coherence values that show areas that respond and really synchronize with breathing. And this is just a plot down here as well, showing kind of all contacts from the frontal, temporal, sensorimotor, and parietal areas and their coherence from 0 to 1 here on the y-axis. You can see that within each of these regions that we have here, superior frontal gyrus, middle frontal gyrus, again, throughout the amygdala hippocampus, you can see a huge variation in terms of their coherence. Some sites have really no synchronization with breathing at all, and some have very significant synchronization with breathing in the frontal lobe, and really, many other key sites that are important in interoception. And those sites that we saw in the insula, again, the amygdala, this anterior cingulate cortex, and even in the sensory motor cortex, which you know is really critical for volitional breathing as well as perceiving breathing, the precentral gyrus. Really all of them kind of synchronize with breathing, kind of some of the highest levels of coherence that we see. You can see this kind of, the green here is LFP and gray is this kind of the respiratory pattern that we have. You can see the frequency is almost very, very similar, and the traces very much overlap. You can see it very, very significantly here, and even the precentral almost completely aligned with breathing. And the LFP and the breathing are almost completely aligned. You can see in this, and we have here is the power, and the power at each of these different frequencies. You can see it has a strong peak at the respiratory frequency here. And at the respiratory frequency, you can see the coherence is very, very high at these compared to, many other frequencies nearby, suggesting again that these are really, really aligned. And maybe this is one of the ways in which our forebrains are able to perceive breathing. And maybe also the way breathing affects the brain in terms of function and emotional control. So we have a bunch of other different experiments we've done with that, and that will be kind of forthcoming. So what can we conclude from all of this is there's really much to learn about how the forebrain controls and responds and perceives breathing. It's really critical for many associated physiological processes. And in turn, breathing really affects cognitive function and emotional control, and understanding that's going to be important. Multimodal studies is going to be important to really advance the field. So I want to give acknowledgements to my lab, and there's many people, but the key members, Ariane has been with you since the beginning, Rakib and Christopher Kovac and Sukhbinder Kumar, who's really been critical in driving a lot of the other research forward that we're doing uniquely, and other things that haven't presented here. Really, this is kind of a multi-institutional, international kind of collaborative effort, though, with many other people. Because as a neurosurgeon, I really knew nothing about breathing control, and it required really the experts in the field and to talk with them, engage with them, discuss our findings with them and thoughts to really help us, move forward and, produce what we think has been, high quality research studies to date. So thank you, and I'll take any questions.
Right, that was a terrific presentation. Thank you, Dr. Dlouhy. I'm going to start with some of the questions here, and I'll remind folks to go ahead and use the Q&A function, drop it in there, though I'll try and retrieve ones from the chat where I can. One of them is a questioner indicated that previously knew that the amygdala was involved in processing emotions, You've hit on this some, but I was wondering what the intuition is for why the amygdala would be involved in inhibiting breathing. You said that breathing affects emotional control. Perhaps you touched on that a little bit, but maybe you can expand on that.
Yeah, yes. Hey, Brian, could you repeat that, I guess?
Yes, I guess, why such a sort of tight link, I guess, between a structure that we know is so strongly involved in processing emotions and breathing. You speculated a little bit on that already, but maybe you can expand on that.
Yeah, I think, yeah, emotion and breathing are tightly linked. Just we know that anecdotally. There's lots of studies on that. And I think that, the amygdala is critical for the fear response, critical emotional control, plays a role somewhat in panic response as well. Um, and part of these responses, the emotional responses of fear and panic, they're defensive responses, and they're protective mechanisms, right? So they're used to, the panic response is a flight or flight mechanism to get away from a predator, and the freezing response is a similar mechanism, right? So it's the step right prior to making a panic response. It's a thinking response. It's thinking, what should I do next? And that the amygdala is critical for that freezing response, and the inhibition of breathing, that's what we think, right, is part of that, is a camouflaging, stopping our breathing, stopping our motion, part of the freezing response, protective mechanism.
Okay, great. There's a question about the breathing synchronization findings. Was there a specific frequency band on the intracranial EEG that synchronized with breathing, and did this band vary across different regions of the brain?
So everyone's breathing is a little bit different. If you just a resting breathing frequency, some people are very, very slow. They'll go from 0.15. Some people are at 0.45. They're breathing 20. So it goes from maybe 8 times a minute, right, to some people are doing nearly 30 times, like 20 sometimes a minute. Just depends on the individual. And with differing frequencies, we saw the power spectrum of the LFPs peaked at those breathing frequencies across different individuals, and we saw the coherence that also was elevated, increased at the breathing frequency at those levels, and it varied across patients. So it wasn't like a specific frequency; it was what the individual's breathing was at. So this is built into their own physiological processing.
There's a question, does there seem to be any effect on the induced apneas under conditions of hypoxia or hypercapnia or hypocapnia? Not the result of the apnea, but the CO2 and O2 levels at the time of stimulation.
So all these patients are, resting, breathing, and they're awake. And we haven't manipulated, CO2 levels. So presumably, most people CO2, they're, 35, 40. In tidal CO2 is 35 or 40. And oxygen saturation for all these patients is, 95 to 100%. And we haven't varied any of that to test it. I'll say it's a good question to ask, though. But we haven't done that.
I have a question for you about the whether or not there are short-term changes in the function of the amygdala that are induced by recent seizure or stimulation. I'm thinking of the fact that patients who have seizure clusters, many seizures in a short period of time, have a higher risk of SUDEP. You showed how the effects of the stimulation or the seizure can linger for a little while, but in a relatively short period of time, they go back to eupnea, to normal breathing, normal CO2 levels. But is there a vulnerability that is conferred if something happens 4 hours later? Do we know that?
Yeah. So for the patients that had this persistent apnea where it lasted, in some cases up to 15 minutes, right?
Mhm.
The in those cases, we looked at the amygdala and just recordings from the amygdala, and we didn't see any differences. Um, and uh, and these patients did not have like persistent seizures beforehand. They didn't have like a lot of cluster activity.
Mhm.
Um, there may be an additive effect. As you're mentioning, Brian, these patients at clusters, they may have a higher risk of SUDEP. So there may be some additive effect. Maybe there's changes within the brainstem that occur also from an additive effect of repeated seizures in the amygdala. And that may also play a role. So it may be something in the circuit rather than just the amygdala itself, but the amygdala brainstem,
Mhm.
central pattern generator circuit.
Okay. Related to that, there's a question based on the time between the amygdala stimulation slash seizure and apnea. Do you think there is a direct connection between the amygdala and respiratory areas, the respiratory network, I suppose?
So sorry, I guess, yeah, say that again. Sorry.
Yeah, as what I guess that you mentioned some of this perhaps in your talk, but is there a what evidence do you have that there's a kind of a direct connection between the amygdala and the respiratory network?
So yeah, so I think we have some data I didn't show here on amygdala connection to volitional areas of breathing in the forebrain, and other critical areas we thought are involved in volitional breathing control. And that's the ESF MRI data. We have resting state data on that from the air site. The connections with brainstem in humans, right, is limited. What we have is the ESF MRI data showing that link.
Mhm.
With functional MRI bold changes with stimulation of the amygdala within brainstem respiratory sites. I mean, that will give you the best, I guess, evidence for it. The apnea is immediate with stimulation onset, suggesting this is, was shut down really of the central pattern generator. How it does that and the circuit it goes through, that's still unknown.
And I think there's also some data as well from some animal models too, including from the George Richardson's lab here at Iowa as well. Um, a question, the chicken egg nature of breathing and emotion seems interesting. Do you see it as breathing impacting emotion processing at the amygdala, the inverse, or both? Or do the two happen in parallel?
Yeah, I think that's still unknown. I think no, we see it as both. Um, and um, but I think, yeah, I still think that so there's other areas, right, that are going to be involved in this perception of breathing. Um, and uh, and I think that's going to be key. Looking at this network, right, of interception of breathing, insula is critical, and many other sites are critical for that. I think that that's still an unknown, but more studies are needed.
Oh. Um, so there's another question here that I will try to capture with the known involvement of the amygdala and the central processing of air hunger. It is very interesting and confusing to see the inhibitory effect on the brain stem. Might it be that the stimulation, being non-physiological, causes an inhibitory effect on the amygdala, that the amygdala is stunned, resulting in no air hunger response and an aberrant inhibitory message going to the brainstem?
Yeah, I think so. We know the amygdala is not required for normal breathing, and so there's patients that have had, and they've done this in animal models, bilateral amygdala lesions, and they breathe normally. Um, the perception of air hunger, I think most would suggest it's insula, and that's critical for it. I'm not sure that that necessarily needs to go through the amygdala. Patients that have bilateral amygdala lesions as well, they can perceive air hunger. There's a great study here by John Wemme at the University of Iowa looking at CO2 hypercapnic, CO2 single 35% CO2 challenge, and patients with bilateral lesions had an exaggerated response to CO2 and had a panic response with increased respiratory rate and perception of air hunger very easily. It was exaggerated. It was greater than normal controls, or even those with panic disorder. So I think that still unknown, I guess, with what amygdala stimulation is doing, although it's likely activating the amygdala to cause inhibition. Just inhibiting the amygdala, I don't think it would have any effect on breathing if that's what the stimulation was doing, since we know that knocking out and lesioning the amygdala has no effect on breathing.
All right, I'm going to ask you a broader question. The research that you do and this patient population, this context, obviously an incredibly powerful, unique access to the brain. What are the biggest challenges in conducting this research?
Yeah, I think that's a great question. I think that the biggest challenge is that there has to be so many things that go right to make this work. So we have a single patient. First off, the steps of it is that they have to, give consent to do the research. So they have to trust you as a surgeon, you as a research team. Mm-hmm. The surgery has to go well. So there has to be no complications. Okay. The patient has to recover very well to do this, right? That they are awake, they don't have much pain, and all that requires, all different things that I do to make sure that happens. So patients are recover immediately. They're on the monitoring unit the next day. They have, good experience. They're no pain, and that they're willing then at that point to participate. So this is both children and adults, right? So in difficult patient population could be at times. And then the experiments have to go well. The recordings have to be good. Electrodes they can't break. Electrode contacts can't break. A lot of equipment's in place. And then you have to have, again, the cooperation of the patient themselves. A great relationship has to be developed. And then that's just to get get data, that's just to get the data. Then you have to make sure the data is high quality. You have to do all the analysis. The artifacts have to be removed, or the patients it can't be included because of their seizure foci. We exclude those, all the sites for that. And it's analysis on the fly, making sure that, okay, do we need to repeat that experimental task? A lot of patients, a lot of hours, many people, a lot of other experts involved in all of this. So manpower is high. The setup that we have here, the hospital, took out a research room, knowing that they'd lose, money, right, to build a research, setup right next is connected to the patient room. So yeah, I mean, I think, it's a huge amount of investment, and from not just from my time, from the whole team's time, from everyone that's involved in this research.
It's a very, very labor intensive and rare, precious opportunities when you do have a subject. I think we have just a couple minutes left, and I'll just see if anyone else has any questions to drop in the chat here. Otherwise, I wonder if you could talk a little bit about sort of what happens. Does anything happen to patients' perception of dyspnea if you end up removing the amygdala as part of the epilepsy surgery?
I think we've done, so we only remove one amygdala. Okay, so that's one thing. If we do the resection, it's only done one. It's only on one side, and the typical perception, the CO2 studies that were done in the bilateral amygdala lesions, it seemed like they had, the response to CO2 was elevated in terms of respiratory response and in terms of the kind of panic response, as well as their CO2, the general panic response and their hyperventilation. removal of one, we did that years ago and see if these patients had a different, like an intermediate effect, and it was kind of mixed results that wasn't really that conclusive.
Mhm.
Although I do think in maybe some individuals it may be affected. We know that removal of the amygdala, temporal lobectomies, they can alter emotional control, and they can have, in some patients they have higher, can be depression, anxiety, even though you're removing the amygdala, and or even panic. So I think that there's probably something involved in that, and probably also not in just like the general regular breathing, but probably involved in some kind of emotional control. Breathing is probably affected.
Mhm. Probably hasn't been studied in great detail,
It
though,
hasn't
right?
been studied in great detail.
Yeah. Okay. Great. Well, that brings us to the end of the hour here. And once again, I'd like to thank Dr. Dlouhy for his really outstanding talk. Also want to thank Jake White and the APS for organizing this webinar, and the audience for attending today and for your questions and interest. Before you sign off, we hope that you will take a few minutes to complete a short survey about this session and your interest in future webinars. Thank you.